Okay, thank you all. I'm Carmina, and I'm going to present my project, MexVar. This is part of my PhD project that I'm doing in the Human Evolutionary and Population Genetics Lab under the supervision of Dr. Andres Moreno in Simvestab. Well, what is MexVar? MexVar is a platform for sharing and exploring biomedical relevant data from the Mexico Biobank. So why did we decide to create something like this? There are four main aspects that we had in mind when coming up with this project. First is that, as Jan explained in his previous talk, we can study biomedical relevant variants in a population in order to, in one part, understand better the genetic factors in that disease, but we can also prove the public health policies that are happening in that population. For example, here, if we see a variant in a population, we can analyze them and have better precision medicine and population-specific medication doses. Also, we can have better prevention campaigns focused on that particular disease. But also, as Jan mentioned, there's this huge bias towards European population, whereas in the time, there's still the majority of the data coming only from Europeans. So with that, populations as the ones that we live in Mexico are not be looking at. So we want to look at populations in Mexico, but in order to do that, we need to have a comprehensive biobank, not only for one region of Mexico. We actually want to capture all the diversity that there is. And for that, the good news is that, as yesterday, the Mexico Biobank project was published. So big congrats to all the people that were involved, to Andres, Machal, Majo, Consuelo. A lot of them are here, and a lot of more of people that were involved in this project, institutions as Simvestab Langevio, Instituto de Salud Pública, Instituto de Nutrición, and this was done with the funding of CONACYT and the University of Oxford. This biobank, as you can see here in the map, each of the dots represent an individual from the biobank. So we have coverage for all 32 states, and we have samples not only coming from cities, we have samples from urban and rural areas. So there's this huge resource for genetics, but we wanted to make it accessible for the scientific community. We don't want it only to be there. We wanted that all kind of scientists will be able to analyze the data and have following the FAIR principles, that the data will be findable, accessible, interoperable, and reusable. So with that in mind is that we create MexVar. And how does it look? So this is how it looks. MexVar is a web-based application where, can you help me start the video? There's this app that you can go, and it will display the map of Mexico, and you can choose one of these biomedical relevant SNPs, and you can look at the frequency in each of the states in Mexico. It will also display you the map, a bar plot with the highest state, in the frequency to the lowest. We show the frequency in a global way, but we also have a novel premises that we're looking at the ancestry. We can separate it and look at the genetic similarity to the indigenous segments and the Europeans. And for that, I'm going to explain more in the next slide. So how do we create this app? So first, we took the Mexico Biobank data. We curate variants that were known to be biomedical relevant. They came from different databases that are specialized in different areas. For example, we have ClinVar, we have PharmGKB for pharmacogenomical variants, we have OMIM and GWAS Catalog. We filter those variants and we calculate the allele frequency, first in a global way. Then we do this ancestry-specific analysis when we take all of the genomes and we separate them into each of the segments that are associated to a particular ancestry. So for example, we can associate a genomic segment to have more similarity to African ancestry. And with that, we can have a mask of each of the ancestries, and we can calculate the allele frequency in that particular ancestry. With that, we integrate all these results into the app. When you can have the input, the SNP, you can show the ancestry that you want to look the frequency in, and also some features as the color palette. And as with output, it shows you the map, the bar plot, and you can download the table in a friendly way. So some of the results that I'm showing here is that we have from all the different databases more than 100,000 samples, 100,000 variants from the different databases. According, ones of them are bigger, some of them are smaller. And here we can see that they have different distribution in the allele frequency. So now I'm going to focus some of the things that we can learn from this database and the results in these allele frequencies. Here I'm showing you only the variants with higher level of evidence in PharmGKB. This means that variants that are known to have an effect in biomedical relevant genes, and there's evidence, there's functional evidence. Each of the dots represents a variant, and in the x-axis we have the ancestry-specific allele frequency for indigenous ancestry, and in the y, in European. So the further point separate from the diagonal is enriched in a particular ancestry. For example, if we look at this sample SNP that is quite far from the diagonal, we see that what this means is that when we look only in the indigenous ancestries frequency, is way higher. It's about 0.7 comparing to 0.1 in the European, meaning that it's enriched in this ancestry. And we can see that this enrichment happens both ways, into the European and in the indigenous segments. And they're associated with different phenotypes, as mentioned with icons. So one of the results, actually, the one that I was pointing out, we can see here the map of this frequency, and we can see a pattern going north to south with increasing frequency in the south states. And when we separate this into the only looking at variants that are present in the European genetic context and in the indigenous one, what we can see is that it's virtually absent in the and Latin American variants. So we can see that in this case, the distribution is based on the genetic background. But also when we see, for example, this variant only in the indigenous segments, we can see also distribution, meaning differential distribution, particularly the Mayan Peninsula, meaning that there's also this differentiation within the indigenous component. These were only two examples of how we can explore this database and what we can learn and the importance of doing these biobanks. And we hope that this tool will be useful for all the scientific community and will help us to bring the gap closer in diversity and help public policies to make more health-related policies focus on the populations. And thank you to all the people in the lab, to Andres and Alexander Ionidis, who are my supervisors in the project, and the lab in general. And all you.
Now we have time for questions. Do we have questions for Carmina?
Hi.
Hi.
It's a great work. So I have a question. So
Yes.
you have mentioned on your slide there are the 100,000 SNPs of the biological reference.
Yep.
So my question is like from where you extracted this information, like what database you used for that?
Yeah, it was for 4 databases: ClinVar, PharmGKB, OMIM, and GWAS Catalog.
Okay, thank you.
Hi, thank you for your presentation. Amazing work, and congratulations for this amazing work. I have a questions about your databases. Moving forward, we know that all these databases are based on effect sizes on European populations.
Yes.
So moving forward, what are your plans to really have Mexican population effect sizes of active SNPs or on our populations? How this database will evolve in the future to account for this?
Yeah, I think I can answer that in 2 ways. First, with the Mexico Biobank, actually in that there were some studies on GWAS run that give us more information that was particular from this cohort. But in the other side, in these databases, for example, in one of the variants that I show, In this one, this has been in PharmGKB. Why that we decide to also focus on this is that there are a lot of functional information about this variant. So we know that this has been worked in several different cohorts, and this variant has been tested in Mexican populations, and it has been shown similar results. So in a way, I think results like this can help us. Well, like the biobank is a result that can give those, but also having this database and showing the discrepancies between the allele frequencies can also help to bring more funding into funding more resource and more analysis with Mexicans.
Thank you. Amazing work.
Thank you.
Thank you, Carmina. So